Good morning, everybody. I hope everyone had a good, meaningful, long, extended weekend. All right, so we will continue our discussions in the introductory series, and this is where I left off on Thursday. Remember, we started talking about control systems, which are a principal component of all aspects of physiology. We talked about intrinsic, extrinsic controls, and positive and negative feedback. Okay, so let's continue that. And throughout the semester, every system that I discuss, there will be control systems, and there will be hemostatic controls. So I will be presenting that usually in some context. This is a classical seesaw technique. If you remember from your childhood, a seesaw is basically you have a big board on a fulcrum, and when it's perfectly balanced on either side, it's parallel to the ground. If it's too much or too light on one side, then you lose that balance, so that homeostasis. So this is a classic way at which we can talk about control systems or hemostatic controls. So the variable is as you see, here's the seesaw. When is it in homeostasis or imbalance? It's parallel to the ground. When there's an imbalance, either too much or too little of that variable, it switches from being parallel. Now, what happens is, to maintain homeostasis, you have to have some mechanism whereby you measure that variable, right? So that's your receptor in the context of physiological control systems. That's measuring the variable. That this is processed in a control center. And what you will find through the semester is that this is where it kind of deviates from engineering base types control systems. It's a lot more complex. In some cases, you have multiple control centers, as well as the receptor can reside in the control center or not. But we'll get into that later when we talk about specific examples. So your control center will act on those changes which are measured in the receptor. That means that we need to affect a change. We need to either increase or decrease that variable. So that means the control center will initiate a effector, which will alter that variable, bringing it back in homeostasis. That's a classic. This is very much a classic. Now, this is just one way of presenting it. If you have some issues with that, here is a more this is an older traditional way where you have a loop system. Again, you have your variable, you have your receptor process, and basically, so in this particular example, you're maintaining blood pressure. So you have two sets of receptors which are measuring pressure. They're baroreceptors. One set resides in the aortic arch. This is as the blood leaves the left ventricle to go into the systemic circuit. It passes through into the aorta. This forms an aortic arch. There's baroreceptors here that measure pressure. There's also a set up here in your carotid arteries that is supplying blood to your brain. Both sets will measure and can act separately to maintain pressure. The ones in the carotid are designed to ensure that you have a constant blood delivery to your brain, whereas the aortic arch is maintaining blood pressure throughout the rest of the body. All right. Now, those are the receptors. Both cases, what can happen is there will be a direct connection to, say, the brain. This will elicit a number of nerve activation steps, which will result in increase in heart beats, heart contractility. It can induce some contraction of certain vasoconstriction of certain blood vessels. The net effect is to increase blood pressure. When this approaches that preset value, you have negative feedback, so you don't overshoot. And therefore, we can maintain this dynamic equilibrium of blood pressure. So this is another way of presenting that. And then the next concept I want to introduce by giving you two certain examples. I will repeat this when I talk about the endocrine portion, but I like to talk about it to introduce the concepts here. One take-home message, just like with your HVAC system, an effector can only change the variable in a single direction. It either increases or decreases. It cannot do both. Just like your HVAC system, you have two effectors. You have a heating unit, you have a cooling unit. You can only use the cooling unit to reduce temperature. You can only use the heating unit to increase temperature. You can't use one to do the other. No different in physiological systems. All right. So let's, and to illustrate that, I am going to use the example of maintaining blood glucose. Right. So you may or may not have heard of insulin. Okay. So let me talk a little bit, maybe pancreas, maybe or maybe not glucagon. So let me talk about maintaining blood glucose. All right. So in this particular example, you go up to Erie, PA. There is a donut shop up there that sells Krispy Kreme donuts. What's unique about this is that when you walk into the store, the aroma of fried dough fills the air. There is a clerk that will poke a hot, fresh dipped donut in your face and give it to you for free. You eat it, next thing you know, you bought 2 dozen of them. That's the whole point. All right. So you set out in your car, because it took you a little while to get there, until you set out in your car, you eat a dozen donuts. Okay. Immediately, you feel a certain amount of guilt. Understandably so. Okay. So the thing about those donuts, they have, and we haven't even talked about nutrition yet. We haven't talked about the carbohydrates, fats, proteins. We will. But basically, let's ignore everything but the sugar, that sugary coating. It's readily digestible, readily absorbed. Within less than an hour to 2 hours, your blood glucose levels start to elevate. They're going up because you ate all that sugar. All right. So your body wants to maintain blood glucose levels. So let's talk about homeostasis and control system regulated toward maintaining that blood glucose. So blood with this increase in glucose is passing through the pancreas. Your pancreas is your receptor. It contains receptor cells that are measuring blood glucose. With this elevation in blood glucose, the control center, which is also residing in the pancreas, has a decision to make. There is an elevation in the variable glucose. We need to reduce that glucose. So the control system will elicit the first effector event, which is to secrete insulin. All right. Insulin is then released into the bloodstream, where it goes to adipose tissue, fat, and skeletal muscle cells. There it will bind to receptors on those cells and tell those cells to take up all this extra glucose that's circulating. Okay, as those cells take up that glucose, what happens to the blood glucose concentration? It goes down. Now, we don't want to overshoot this, so as the blood glucose level goes down, that means that we then reduce the amount of insulin that's released. So those cells are not taking up as much. So that's how we can take up this elevation back down, but we don't want to go overshoot. So we have this negative feedback. All right. Now, you had a dozen of those Krispy Kreme donuts. Those various cells in your body can only take up so much. What happens to the extra? Evolutionarily speaking, you didn't eat 3 meals a day. Okay. You may eat today, you may not eat till next week. So your body has evolved in order to store extra energy and nutrients when they're available. Okay. So what happens is, because you had all that extra glucose, your fat cells and your muscle cells can only take so much. So the body's going to store some more of that bulk. It's not going to waste it. So what basically it's converted to it goes into the liver, where it is converted to glycogen. Okay. Now it's just stored. Think of it as banking it. Okay. Now it is storing that. Now you feel a certain amount of guilt for consuming a dozen Krispy Kreme donuts, right? So you swear off food for 2 days. You make it for 3 hours because what happens is you get this big peak and then it goes back down. Oh, and you're not eating anything, but all the cells in your body still need sugar to work because it's a primary energy source. So what happens is now your blood glucose levels drop down because the cells are utilizing it, but you're not bringing anything in, dietarily speaking. So what happens is, again, the reduction in blood glucose is again measured by receptors within the pancreas, a different set of cells, and the control center, which is also in the pancreas, decides that, oh, we need to elevate blood glucose levels. So it will release a different effector called glucagon. Glucagon goes to the liver and mobilizes those glycogen stores. And then the liver converts those to glucose. They're released into the blood. Blood glucose levels go up. As they approach their preset value, there is negative feedback, so we don't significantly overshoot. All right? So we can use the seesaw technique here to illustrate, but the point of this discussion is that an effector can only regulate in a single direction. You have to have multiple effectors in order to make it go up or make it go down, right? So all you can do in one direction to protect from overshooting is to reduce that effector so that you reduce that effect, whether it's increasing or decreasing the variable, all right? You gain additional control by having eliciting multiple effectors which can direct that variable in different directions on top of that. And this is just an example within your body. It's probably one of the best ones that I can use. All right. So working on down. Again, this is another way we can have these chart bubbles where, for instance, in this regard, we're doing is maintaining body temperature. Remember, I mentioned in last week, your homeotherms, you maintain a constant core body temperature despite changing environmental surrounding temperatures. In this particular instance, in this example, you have your temperature starts to go up. Maybe later today, it's not going to get that hot, but let's say it gets a little bit warmer. Your body starts to, it's retaining heat more because there's less of a temperature differential between you and the outside. Your body temperature goes up. This is measured in a set of receptors in your brain. And they will then initiate a series of effectors with the net result being to reduce it. These effectors can be such things as inducing you to sweat. This is going to increase evaporative heat loss. And associated with this is increased circulation, delivering blood to the surface, to the skin surface for either conductive loss, radiant heat loss, or evaporative heat loss. The net effect of all of these is to reduce temperature and bring you back down. And then, of course, as you maintain your core body temperature, you minimize those effector effects, okay? So all of these are examples of control systems. And I will be giving you control systems all throughout the rest of the semester. Now, up to this point, I've talked about negative controls. Negative feedback, whereby we want to stay within a certain physiological state. Remember, when I talked about this on last Thursday, I said the primary difference between positive and negative feedback is a negative feedback is responsible for keeping you within a given physiological state. The point of a positive feedback is in order to transition from physiological state A to physiological state B. Now, let me give you 2 examples. The first one is that your blood is flowing through your circulatory system, and if you cut yourself, what happens? It clots eventually, depending on how the severity of the clot and which vessel you cut. Of course, if you cut your carotid artery, forget it. So, but we're talking, you're cutting up dinner tonight and you nick your finger while you're chopping up your veggies, right? So, you can do a couple of things, but generally speaking, if you don't do anything, it will eventually stop and the blood will clot. The purpose of that is to keep you from losing blood. because blood is a critical asset for you. All right. Now, we'll talk about the details when we get into the circulatory system and how this works. All I want you to realize at this point is that you have a hole in a blood vessel. You're losing blood. Okay. That's going to initiate a cascade of events that will essentially result in closure of that hole. Okay. So the point here is this is an example of positive feedback. So we have two physiological states: bleeding, not bleeding, whole, no hole, right? We do not want to maintain homeostasis, in other words, continually losing blood. We want to stop that. So what happens is when you have think about your blood vessel and the cells that line that blood vessel, think about them as a Teflon coating, okay? Now, blood contains both a liquid called plasma and a cellular portion. The mixture of this is flowing through all of your vasculature all of the time. With this Teflon coating consisting of these endothelial cells, the blood just flows through. Now, if you have an injury, that's going to strip away some of those endothelial cells. The material that lays underneath is such that with the removal of this Teflon coating, certain cells in the blood bind to this underlying surface. This initiates a cascade of effects. There's multiple effects. The net effect is one increases the next one, that increases the next one until we get a blood clot or a closure of the hole and we don't bleed anymore. Okay, so that's positive feedback. Transitioning from physiological state 1, bleeding, to non-bleeding physiological state 2. A second example, when we get into the reproductive portion, we'll talk about various, especially in the female reproductive system, we will talk about an estrous cycle, okay? And that consists of a follicular phase and a luteal phase. During the follicular phase, there is the development and maturation of the egg, such that it is capable of fertilization by a sperm. This resides in a various structure called a follicle. And it's only called a follicle so long as there has an egg within it. At the middle of the estrous cycle, the egg will leave the follicle, and it is no longer a follicle because it does not contain an egg. And it is now called the luteal phase, and it has a different function. Now, so the whole point here is we have to transition from the follicular phase to the luteal phase, right? Now, how does that work? All right. So we need to have ovulation. We need to lose the egg. So how is this directed? Toward the end of the follicular phase, there is estrogen being produced. All right. Now, more estrogen it's going to be produced and released from the ovary, from the follicle. It's going to go up here to the brain. Don't worry about where in the brain, but what's going to happen, the brain is going to release a hormone called luteinizing hormone, LH. LH goes to the ovary, promotes the release of more estrogen. With positive feedback, that means more estrogen, more LH, more estrogen, more LH. Eventually, we get a spike of LH, which induces ovulation and loss of the egg. So this is another example of positive feedback. And we are transitioning between follicular phase and luteal phase, phase 1, phase 2. You don't want to be stuck in the middle. Without that LH spike, you cannot have maturation or release of a mature egg. And when we get into the reproductive facet, we'll talk about the basic classic pill, which actually stops this process, okay? But we'll get there later, all right? I'm just introducing a positive feedback. All right, now, let's leave that section. Remember when I introduced the introductory lecture series, I said I'm going to be talking about a range of different things. And the whole point of this is to bring us up to a common level before we get into a discussion of the individual systems of physiology. Okay? So now I want to focus on cell biology. All right. Now, don't worry. This is not a class in modern cell biology. However, there are critical concepts that I do want to introduce. Because if you do not understand what is a cell, how does a basic cell function, especially the barrier membrane of that cell, we can't talk about physiology. All right. So first off, cell theory. Okay. This will be old for some it won't be. So basically, all living organisms consist of cells. You have single cellular organisms, which are a single cell, or you have which are trillions, tens of trillions of cells, right? Now, number two, within your body, all of those cells provide a structural and functional aspect of the body. You cannot be you without these living cells. They create products, which then those cells and their products make you you. And you can't be you without those cells. Now, next, all of those cells came from a single cell that fertilized ovum. That's how your life began. Now, there was a series of mitotic divisions, and eventually, mitotic divisions, specific differentiation, now we have you. So all of the cells in your body that are part of your body directly came from that cell originally. Now, let me introduce here is those are not the only cells in your body. Okay? Those are the cells that make you. You've heard of the term microbiome. Everyone's heard of microbiome. So microbiome are those cells or cellular organisms, most of them single cell organisms, or in the case of viruses, not even cells, that reside in your body, either in your gastrointestinal tract, on the surface of skin, in your ears, your respiratory tract, wherever. So those actually, if we divide that into cells, that actually represents 100 times the cell number of those 75 trillion cells which make you. Okay? But that's a different story. Let's focus on those original cells that all derive from that fertilized ovum. So those cells came from this cell originally. All cells have to come from a pre-existing cell, right? Now, because those cells share a common ancestor, they have certain properties that are unique and represent that. Okay, we could go all the way down to the DNA, but we won't at this point in time, right? Okay, why all these cells? Why not just have a big cell, be big amoeba? No, it ain't going to work for us. There are size constraints, and it all has to do about transport. All right. So there is a limit to cell size. Now I'm going to walk through and give you give you an example. But let's talk about, first off, what are the size of cells in your body? Most of the cells in your body, if we reduce those to their least expensive energy concept, they're a sphere, they would be 10 to 15 microns. Most cells, over 98% of the cells in your body are that size. A couple of exceptions. An egg is the largest cell in the body, and it can be 100 microns. Next, we have neurons, which can be the longest cell in the bodies. I'll talk about them in more detail when we get to the nervous system, but if we talk about a motor neuron that begins in my finger here, my index finger, it goes all the way up my arm and goes to my vertebra here to interact with the central nervous system. That's a meter long. So they can be that long, but they're not very big around, right? So why are the cells this size? Okay, it all has to do with surface area to volume and transport. It all has to do with transport, and that's a function of surface area to volume ratio. And basically, the thing to remember is that as I increase size, I disproportionately increase volume in comparison to surface area. So don't worry about that. Let's give an example. I like to work through this. So in this particular example, instead of a circle, let's work with a cube. Okay. And it's just for keeping the math simple. So we have two sizes of cells. We have a traditional size of a cell, which is 10 microns. That's the smaller cube. Okay. So it's 10 by 10 by 10. Okay. So its diameter is 10 microns, right? Its surface area is 10 by 10 by 10 by 10 by 6 because there's 6 sides. That gives us 600 microns squared. And if we do volume, basically it's 10 by 10 by 10 or 1,000 microns cubed. Okay. Now, let's just simply double the diameter of that cell. That has a disproportionate effect. So we are only doubling the diameter. So that means that the surface area is 20 by 20 by 6. That doubles, a little over doubles the surface area going from 1,000 to or 600 to 2,400. But look at volume. So that's 20 by 20 by 20. That's 8,000 microns cubed. So there's this disproportionate increase of volume versus surface area. And this all has to do with transport. So that makes it harder to get things in and to get things out of that cell. All right. So this is one of the reasons why we don't have millimeter-sized cells in the human body, because transport becomes very, very difficult, right? Now, just what is a cell? We have trillions of cells. We have different kinds of cells in the body, and they do different things. Now, I'm giving you an example of the anatomy inside a cell. First off, don't panic. I don't need you to memorize the anatomy of a cell, and this is fairly simplistic. I'm just introducing the complexity, and this is relatively simple. The first thing you have to notice, and I want to denote a couple of things. First is that the outer aspect or the membrane of the cell forms the primary boundary that separates the cell from everything else. Okay? So that means that separates the cell to what's inside and what's outside the cell. So in order for any transport to occur, it has to cross that plasma membrane, either going in or going out. We will spend a fair amount of time talking about that membrane, but right now let's focus on some other parts. In the center of this cell, more or less, you see that purple-looking thing. That's a nucleus. This contains the DNA of the cell for the most part. And whereas every cell will have a membrane, not all cells in the human body will have a nucleus. There are cells in your body that do not have nuclei. So your mammals, your red blood cells, your erythrocytes do not have a nucleus. It has been argued that for a long time, for decades, it was argued that, well, this increases the efficiencies of cells passing through the capillaries. This allows them to pack in more hemoglobin so they can carry more oxygen. Well, this was just disputed a couple of years ago. We still don't know why, but we don't have them. If you're a chicken or a dinosaur, you have nucleated red blood cells, and you have this similar transport efficiency. Okay? But in humans, you do not have a nucleus within red blood cells. Next, there's another cell type. Those are called platelets. When I was sitting where you are, I was taught that platelets are cell fragments. However, my mind has been changed in later decades. Now, the general consensus is platelets are just very small cells, but they also do not contain nuclei because they couldn't fit. Okay? So those are two examples of anuclear cells. Now, there are also cells in your body that contain more than one nucleus. So we will be talking about muscle cells, skeletal muscle cells specifically, have numerous nuclei. You also have an osteoclast, which occur in bones, and they have multiple nuclei. Macrophages can evolve to have multiple nuclei. So those are three examples of multinucleated cells. So even though I'm showing you a single nuclei, that's most cells, but not all cells. All right, there are also Golgi apparatus. There's mitochondria, which are the powerhouses of the cells. Originally, a mitochondria formed evolutionary way back when a bacteria fused within our system, and they actually contain different DNA. Okay, so that's one way, and that's one how we can trace maternal descent, because the mitochondria are they're not part of the sperm, but they're part of the egg. Okay. So, but that's another story. So we have mitochondria. So these are just some examples. The only things that I want you to focus on are what I talk about later in the semester. This particular chart talks about why we have them, what are the functionalities of those organelles. You can read that at your own convenience. So, enough of the generalities. Let's zero in on the plasma membrane. That's what I want to really talk about for the rest of this series. Okay? Because again, this is the interface between the cell and the outside world, and it is the interface of which things have to cross, either going in or going out of the cell. So we need to understand what is the plasma membrane. All right? So it is creating a cell boundary. It controls the interaction of cells interacting with anything outside of the cells. And if we look at, in this particular example, here is a TEM showing this dark double line here. This is a plasma membrane. What is on the outside of the cell, or the extracellular space, is on this side. Inside is called the cytoplasm. That's the inside space, inside the cell. This here is a nucleus, and it has its own membrane. But let's focus on this plasma membrane here. Okay? Now, in the plasma membrane, part of that membrane, it's a bilayer. Part of it is facing outside of the cell, part of it faces inside the cell. Okay? And basically, it is comprised of phospholipids, fats. Okay? These fats are hydrophobic, and they like to be associated with other things that are hydrophobic, and they are repelled from water. You have water on the inside of a cell and water on the outside of the cell. All right? So let's talk about the next thing is the fluid mosaic model. Again, I'm old enough to remember when this was just proposed in the early 70s, late 60s, early 70s. It is no longer new. It's now fairly established science. But basically, what you see here, notice these little balls. These are the heads of phospholipids. They are basically hydrophilic. That means they're attracted to water. The tail portion here, the double tails, these are hydrophobic. They are repelled from water. So what happens is they self-associate based on this hydrophobicity, hydrophilicity. So all of the balls, the heads, are directed either toward the outside or the inside. The tails are equally repelled from water, so they tend to be attracted to themselves. So now what you have is you have this bilayer of phospholipids, and the heads are toward the outside, the tails are toward the inside. Now, there's nothing holding this together except the relative hydrophobicity, hydrophilicity. So basically, if you've ever been to carnival, and I don't know if they had it in recent years, I haven't been to a carnival in many years. So there used to be a little tank, and you would have these ping pong balls floating in the tank. And so, or just imagine a tank, or those little rubber duckies. Okay, so they're sitting in the water. What do? They float on top. They don't go down. They don't leave. They stay on the surface, right? Now, if you disturb them, what happens? They always return neighbor to neighbor. So if you put your hand in those between those ping pong balls and pull it out, what happens? It self-heals. It's exactly the way your plasma membrane works. Those phospholipids are constantly moving, but they can only move in a single direction. They can only move in the XY, not in the Z direction. Up or down. They can only move this way. Okay? But they're fluid. That's where the fluid aspects of this title comes from. Right? Now, interspersed in these phospholipids, there are proteins. Okay? And these proteins can be transmembrane, that is, they can extend across both layers of phospholipids, or they can be stuck on the top or whatever, and then they can also be anchored by the cytoskeletal elements on the inside of the cell. But we have phospholipids, which is the fluid part, and then we have the mosaic part, which is these proteins, which are interspersed within this plasma membrane. So we have the fluid mosaic model of the plasma membrane. Now, those proteins can be anchored via filaments, or they can just be floating around, but they are locked in because in order to be a transmembrane protein, this part is hydrophilic, but the central portion is hydrophobic. So it can't leave. It's trapped here, but it can still move around, right? So that's where the concept of fluid mosaic model of the plasma membrane comes from. This is important, and you need to realize this. And basically, well, wait a minute. I can understand the concept of phospholipids. They enable a barrier. They create the basic aspect. They create the bottle. That is the plasma membrane. Why do we have proteins? What's the role of proteins in this fluid mosaic model? All right. Well, let's get there. So proteins only make up about 2% of the molecules found in the plasma membrane. The overall bulk are your phospholipids. However, because of their larger size and weight, they comprise up to 50% of the plasma membrane. Right? Now, why do we have them? So we have the integral ones, which I just talked about, where they are locked here because of the hydrophobicity here, but these are hydrophilic here. These can anchor to intracellular cytoskeletal elements. On the surface, they can bind to any number of things. Okay? So we have proteins. They not just proteins, they have glycoproteins, and those are proteins with sugar elements, sugar molecules on them. And basically, we have these kinds of proteins. So we have proteins passing through transmembrane or integral proteins, and they can be on the surfaces. Right? So what are these proteins doing? They're there for a purpose. Matter of fact, they have a number of potential functions. Let me go through this because these are important. So remember, before I start with this discussion, your phospholipids create the barrier. Proteins allow you to cross that barrier somehow or another. Because remember, if you have information on the outside or some molecule on the outside that you have to get on the inside, depending upon the nature of that molecule, it has to pass through that lipid bilayer. If it's not hydrophobic, it can't. All right. So let's talk about. So this is kind of a segue into what are these kinds of proteins that are associated with the membrane. So the first one is transport. All right. So we'll talk about more detail about the membrane in a moment. But basically, let's say that you have a molecule, say a sodium ion. It has a positive charge, which means it is not soluble across the phospholipid bilayer. But you need to get sodium into the cell. So, well, how do you get it in there? Well, these proteins, some of them can be transport proteins, which transport or facilitate the transport of sodium into or out of the cell. Okay, so that's the first element, transport. Number two is enzymatic activity. All right, so especially on the internal or intracellular side, you can have these proteins associated with have enzymatic activities so that they can be activated for enzyme activity. So that means they're breaking down stuff. All right. They're also receptors for signal transduction. Right. So wait a minute. We'll get into the details when we get into the endocrine system. But let's say I've got a protein signaling molecule. Proteins tend to have a charge, so that means they can't pass through the plasma membrane by themselves. But they have to signal the cell. They can't get into the cell, but how do they signal the cell? Well, they dock up to proteins that are on the surface of the cell. And those act as receptors. Here's the signaling molecule. Here's the receptor. It binds to the receptor. There's this binding interaction. There's a transmembrane protein that will then pass that signal into the cell, right? So we have transport, we have enzyme activity, we have receptors. We also have something called intermembrane joining. So your skin is a barrier. It's your primary barrier, right? You have the epithelial cells that are prime constituents of that skin. These epithelial cells basically are glued together. The glue that holds these cells together are proteins. These intercellular molecules holding them together means that the water or things can't really, because things can go between the cells, but if they're really tightly joined together, it forms a barrier and it keeps things from going between the cells. And that's one function, is intercellular joining. Cell-to-cell recognition. Here's cell A, here's cell B. Like cells tend to self-associate. How do they self-associate? If it's a lipid bilayer, hey, it's a lipid bilayer. There's nothing unique about it. But if you have different proteins on the surface and they are designed to recognize each other, that means here's cell A, cell B. There's this common modality. They will then link up and join. So that is the next one. All right. So cell-to-cell recognition. And the final one, so cells exist. They can be two types of cells, basically, in the body. You have cells that are just floating, like red blood cells. Okay. Or you have cells that are in tissues. Those cells don't float. Those cells have to be attached to something. That's the extracellular matrix. So how do cells bind to the extracellular matrix? Not by the phospholipid bilayer, but by proteins that are inside, are on top of that surface. Those are called, generally speaking, integrins. These are sets of proteins which then can recognize various extracellular matrix molecules. All right. So we have cells that cell membranes have proteins associated with the phospholipid bilayer. They have 6 general functions. One is transport. Second is enzyme activity. Third is receptors. Fourth is intercellular joining. Then we have cell-to-cell recognition. And lastly, we have attachment to the extracellular matrix. So these are the reasons why we have proteins in this fluid mosaic model. They allow for the cell to have transport. They allow for communication both inside and outside the cell. They also allow cells to recognize other cells, etc. So they have a multitude of functions. And we'll talk about those. Okay, so let's talk about cell-to-cell direct communication, okay, or cell recognition. There are 3 things I want to talk about: tight junctions, desmosomes, and gap junctions. All right. Now, and they all have to do with cell-to-cell directed communication. So I'm used graphically to talk about it. So let's first talk about tight junctions. Now, tight junctions, I gave you the example of the skin. That's what tight junctions do. They create really tight joining between 2 cells, so things have difficulty passing between them. All right. Now, if you want to think about them as glue dots, if you've worked with glue dots in crafts and so forth, they hold things together very tightly. Bless you. That's what happens here. All right. So you have the corresponding blue dots on two adjacent cells. They dock up. We form a tight junction. Bless you. And depending upon the density, this makes it extremely difficult for things to pass between those cells. All right. So tight junction. The next thing I want to talk about is something called a desmosome. A desmosome. I want you to think of a desmosome as Velcro. Everybody knows what that is. You've got your loops and hoop structures. Here's the hoops, here's the loops, and they hook together. You can pull them apart, but they tend to hold together. So in the desmosome, that's what these linker proteins serve as. That's the Velcro. They're anchored on these plaques. So look, these linker proteins are exposed outside of the cell, but they're anchored inside of the cell to these plaques. Now, not only are these linker proteins embedded in these plaques, there are basically also these intermediate fibers that are also anchored to the plaques. So here is cell A, here is cell B, here are the linker proteins, they're interjoined, right? What happens if I stretch cell A, that's on my right here? If I pull this cell, what happens? If I pull that cell, that's going to pull those linker proteins, which are attached to the plaque, which then are attached to the linker proteins. And what happens is they're going to pull it this way. So as I'm pulling the cell this way, I'm putting tensile strength on the linker proteins in the adjacent neighboring cell to its anchor point, to its filament, to its intermittent filaments. And so I'm going to stretch those intermittent filaments. So this allows from mechanical communication between two adjacent cells. Those are roles of desmosomes. The next are gap junctions, right? Gap junctions basically are like creating a hole between two adjacent cells, all right? This is enabled by these connexins. These are a group of proteins. Notice that each of the two neighboring cells have them. If they line up, you will create a physical channel between two adjacent cells. So that means anything that can fit through that hole, fit through that channel, can be freely exchanged between each of these neighboring adjacent cells. The limitation is this. That space is pretty damn small. So only particles or molecules that are less than, say, 0.3 nanometers or something, very small, salts, amino acids, things like that can pass through, right? But you can have direct exchange between two neighboring cells. Okay? What are the advantages? Why? Okay? So without connexins, that means that in order for this cell to give that molecule to this cell, it has to be released into the interstitial space. It's subject to dilution in that interstitial environment. Then it has to find a way into this cell. It's extremely inefficient. Direct transfer assures no loss. It's a complete, from an efficiency standpoint, it's highly efficient. There's no dilution effect, okay, in the interstitial environment, since it never enters the interstitial environment. It's direct cytoplasm to cytoplasm transfer, okay? That's gap junctions. All right, with that introduction, I've kind of alluded to this, but let's back up for a moment and talk about membrane transport, all right? So I've already said things have to go in, things have to leave the cell. That means they have to cross this plasma membrane, all right? So certain things can be permeable, which means it can cross that membrane directly. That means it can fit between those phospholipid bilayer molecules. If it's impermeable, that means it's repelled. It can't go by itself. It cannot cross the again, the phospholipid bilayer. If it is selectively permeable, that means it can, some things can pass and some things can't pass. Your plasma membrane is selectively permeable because some things can pass through directly and some things cannot pass through directly. Okay? So basically, it's a function, transport is a function of two things. Again, focusing on the phospholipid bilayer. It's a function of its hydrophobicity, or just the opposite. So that means that if it is soluble in lipid, which means hydrophobic, it can pass through between those phospholipid bilayers. So it can directly be transported. But now becomes the second aspect. Not only is it soluble in lipid, how big is it? The bigger it is, the harder it is to pass its way through. So size and hydrophobicity are the two aspects that will determine whether it is permeable through the plasma membrane directly, right? Now, what are examples of things? So basically, anything that is fat or lipid based, I can just pass by. Again, a function of size. The bigger the molecule, the harder it is. So things that are nonpolar, that is oxygen, CO2, fatty acids, can freely just cross the barrier too, the phospholipid bilayer. All right. Charged molecules such as sodium, which I talked about a little bit ago, potassium, which is also a positively charged ion, as well as glucose and proteins don't. Okay. And I've already told you what helps to them are these transport proteins. Okay, so let's move into that. So again, this is just bringing everything together in terms of membrane transport. Right? Now, protein channels, these proteins that form channels usually are very small things, like they're facilitating the movement of sugars or charged particles and things like that. And again, the way things move, if they are permeable, either through direct permeability across the plasma membrane or the transport, what determines the direction of transport is based on what is the concentration difference across that membrane. Okay, we're going to talk about diffusion in a little bit, but basically that means if you have a stronger concentration on the outside and a less concentration on the inside, and the membrane is permeable, what direction will the transport occur? You're going to go from higher to lower, so you're going to go from outside to inside. If we reverse that and say you have a higher concentration on the inside versus the outside, and it's permeable, you're going to lose, the cell will lose those molecules. All right? Now, well, wait a minute. What if I want to concentrate something either inside the cell or outside the cell? That means I'm moving against its concentration gradient. So that means in order to do that, I have to expend energy. So that in the aspect of the body, that means I have to expend ATP. Okay? So aside from things that are freely permeable across the plasma membrane, let's talk about transport, facilitated transport. This means that it's facilitated by proteins, these intramembrane proteins. And so let's spend a little bit of time talking about that. We also refer to this as carrier-mediated transport, because without these mediating proteins, you do not have transport of these particular molecules. So first off, you have to have these protein molecules that will facilitate transport. Now, the thing about facilitated or carrier-mediated transport is we're talking about individual molecules being moved. If we are talking about larger things either particles or fluids and things like that, we call it vesicular transport, right? And I'll talk about both. Now, let's start by talking about carrier-mediated transport. And regardless of what's being transported, there are a set of rules that go across everything. So the first one is specificity, is that you have these carrier or transport proteins. They are specific for transporting a specific molecule. So you have one set of proteins that transports, say, sodium, another one that transports chloride, or maybe sugars or something like that. So you have specificity. So that means that you have the transport proteins that will be distributed across the surface of a cell. You have different proteins which are designed to transport different things. That's specificity. Next is saturation. Remember, you have a fixed surface area on the cell. A fixed surface area. That means that it's populated, 2% of that surface area is with these proteins. Not all of those proteins are transport proteins. They serve other functions too. But let's just deal with those transport proteins. You have a variety of transport proteins, but they each will be responsible for transporting a single molecule. So that means that with this fixed landscape, I have a fixed number of transport proteins for molecule X. So that means that there is a limit to my transport because the transport is only enabled with those specific transport proteins. I can reach saturation, whereby if I continue to increase the concentration difference, if I have got all of my transport proteins already engaged, I've reached saturation. So I cannot have any additional transport. All right? So that's saturation. Lastly, competition. Now, well, wait a minute. What happens if I've got molecule A, molecule B? They share a certain amount of chemical complexity so that they are more similar than dissimilar, so they have the ability to bind to their respective transport proteins. So that represents competition. Okay? So all transport proteins exhibit specificity, saturation, and competition. Now, let me give you an example of saturation. So if we look here on the x-axis, we have the concentration difference between outside and inside the cell. And that means that of the molecule that's being transported, as we increase the concentration on the outside of the cell versus inside the cell, we are going to engage more transport proteins, move more molecules from outside to inside the cell. Now, as we increase this concentration difference, we will get a corresponding linear increase in overall transport. However, once we've reached saturation, we no longer can get a continued increase in transport because we've already engaged all of our transport proteins. So we level off at something called our transport maximum, right? And this is due to saturation, right? So even though I continue to increase the concentration difference between outside and inside the cell, once I reach my transport maximum, I know I cannot further have any additional increase in transport. I've already engaged all of my transport molecules. Let me give you an example, an analogy to explain this. We have a village. It's a very poor village in a jungle. Okay. The village has about 100 adults. Now, again, it's a primitive village. Everything's made from the surrounding jungle. So they live basically in huts made of the local vegetation, highly burnable, highly combustible, right? So there's nobody there to help them except everyone that's in this little village. This village has 100 adults. All 100 adults comprise the volunteer fire department. All right. Now, it's an extremely poor village. So even though they have 100 constituents for this volunteer fire department, they only have 10 buckets. Now, the problem is, in order to put water on a burning hut, you have to take a bucket, put it at the river, pick up the water, take it and throw it on the hut. All right. So here is sometime in the middle of the night, someone yells, fire. So the first person gets up, grabs a bucket, goes to the river, starts to put water on, right? Second person goes and put water on. That's doubling the delivery of water to that burning hut. Third person triples it, so on and so forth until you get to the 11th person. We only have 10 buckets. So despite the fact that I've got 90 more members of this volunteer fire department, I only have 10 buckets. So I have reached my transport maximum. Okay? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Even though I have 90 more people, they can't do anything because they don't have any buckets to transport the water. So they have reached their transport maximum, right? Now, for this carrier-mediated transport, in addition, I want to talk about, we can divide it again into, does it require energy for transport or not? So the first one is facilitated diffusion. So the only thing that drives facilitated diffusion, beyond obviously saturation, is the concentration difference. As I increase the concentration difference, I'm going to increase transport until I reach saturation, of course, right? So it doesn't require energy. It requires no energy input. There is a concentration difference. It's going down the concentration gradient, whatever. The next one is active transport. In active transport, basically the way to think about this is you're going against the concentration gradient. So in other words, you have a higher concentration outside versus inside, but the cell wants to get rid of stuff. So it can't use facilitated diffusion to go from inside to outside because it can't go not without expending energy, because it needs to pump that material out of the cell, right? Secondary active transport is where you will expend energy to move molecule A and you piggyback molecule B along with it. So basically think about transporting two molecules for one. All right. So let me give you an analogy here. We have a summer camp. Okay. There's no running water, but they have a shower. The shower consists of a bucket with holes in it, and then you pull on the rope, and that opens and allows the water to go through. So how does it work? You take a bucket of water, you climb a ladder, and you pour that water into the bucket that actually forms the shower. Right? So that's wonderful for the first person who takes a shower in the morning. They just get up, they get in there, pull on the cord, and they have their shower. Now, it's a big 10-gallon bucket, and they are lazy and they like their shower, cold water though it may be. All right. So they hold this, and all 10 gallons goes out on that one person. And they're a slob. And so what they do is they just go their merry way. All right. There was no energy expended other than to pull on the cord. Water came down via gravity, right? Fine. Second person comes on, pulls on the cord, nothing happens. But he wants to have a shower. So what does he have to do? He has to go and fill a bucket, climb the ladder, pour it in the bucket, then he can have a shower. So he has to expend energy such that he can get that water. So that's the same thing. So you're actually that's where you have to you don't have a gradient, you're going against the gradient. So you have to expend energy. So active transport and facilitated diffusion. So graphically, let's look at it this way. So we basically are going down our concentration gradient. So that can determine directionality, and that determines rate of transport. The greater the difference, the greater the transport. Okay. Active transport, perfect example is when we have active transport. We'll get into the details later in this system, later in this series, but basically the cells need to maintain more potassium inside and more sodium outside. Now, there is a constant movement of the respective molecules either in or out of the cell. So they're always wanting to keep equilibrium. So in order to keep the difference, that means that we need to pump sodium back out of the cell and potassium back into the cell. And since we're going against their respective concentration gradients, that means we have to expend energy, hence active transport. Secondary active transport. In this example, we are expending energy to transport sodium. Oh, and by the way, we will co-transport glucose. So we're expending the same amount of energy, but we're moving two molecules. And this is called secondary active transport. Okay. Let's talk about vesicular transport. What is that? This is moving larger things, including fluids. All right. So first off, we will divide vesicular transport into which direction is transport occurring. If transport is going from inside the cell to outside the cell, it's called exocytosis. If it's going from outside to inside the cell, it's called endocytosis. Right? Now, the next thing is there are several concepts. So we have for something called phagocytosis, which occurs by immune cells. And what they're trying to do is grab large particles. These could be a grain of sand. This could be whatever particles. It could be a bacteria. And they are grabbing it and internalizing it. All right. You can also have this concept of penocytosis. Think about that as cell drinking. You're bringing in liquids. And then we have something called receptor-mediated transport, which means that endocytosis is directed by receptor-ligand interactions. Now, I'll give you a graphic representation of those. So, in terms of exocytosis, we have something called a secretory vesicle. Notice that that secretory vesicle has its primary boundary layer being a phospholipid bilayer. Think about that as a little bottle, a little jar that is made by its own set of phospholipids. Now, this is inside the cell. Here's our secretory vesicle. Here's our membrane. And there's stuff on the inside, the luminal surface. There's a liquid. There could be proteins. There could be RNA and DNA. It doesn't matter, but there's cargo inside. All right. The goal is the cell wants to get that cargo from the inside to the outside via exocytosis. Without going into all the cell biology details and molecular biology involved, basically there's transport of the secretory vesicle to the internal side of the plasma membrane. Phospholipids of the secretory that form the membrane of the secretory vesicle, again, interacting with phospholipids of the membrane. So they fuse. All right? Now, notice that these phospholipids are fused, and then with the inner side, and then we have the outer side. This is the inside. This is the outside of the secretory vesicle phospholipids. They fuse on the intracellular side of the phospholipids. Eventually, what happens? This then exposes the internal layer of phospholipids. They intertwine with the outer phospholipid bilayer. This creates an opening whereby what's inside the secretory vesicle is now released into the external surface, external volume, the interstitial fluid. Okay? there's something else that happens. What also happens, note that there's this fusion happening. So this is one way. Exocytosis is not only permitting the transport of what's inside that secretory vesicle to outside the cell, it's also replenishing the plasma membrane, phospholipids. Okay? So there are constant loss of phospholipid from the plasma membrane going out via exocytosis or coming inside via endocytosis. And this is one way at which the cell maintains a balance. So this can replace phospholipids that are lost via, say, endocytosis, which we'll talk about in a moment. Okay, so within endocytosis, here's the example with the phagocytosis. So we have an immune cell. It encounters some sort of a particle, whatever it may be. It engulfs it. That means it's surrounding this material. And then this is an internal pinching. So now we have phospholipids that were originally part of the plasma membrane. They're now in this phagosome. And then this is internalized and this can be processed. Okay, so this is endocytosis, phagocytosis specifically. Next, we can have bulk phase phagocytosis. So what happens here is there's an interdigitation of the plasma membrane, and this is going to incorporate what is in the interstitium fluid here. Then it's going to pinch off and now have a membranous vesicle, and it contains elements that were on the outside, now on the inside, and now inside the cell. The next one is receptor-mediated endocytosis. This is endocytosis, but it is specific because it's associated with membrane ligand interactions. So in this particular example, there are receptors, and these are proteins. Remember, I talked about that. They then will be docking up with other proteins because these molecules that form the little triangles have to be hydrophilic. That means they can't pass through here, so the only way they can communicate is binding to these surface receptors. Once we start to get a saturation in this local environment of these receptor-ligand interactions, this will engage or initiate receptor-mediated endocytosis, because surrounding here we have these clathrin-coated pits. Because we have separation, they will induce interdigitation in the formation of this vessel. Okay, since this is directed by a ligand receptor interaction, it's referred to as receptor-mediated endocytosis. Okay. This particular slide, I'm not going to go into it, but this is everything I've been talking about for the last 15 minutes. All right. This is talking about transport and what is transported, what isn't transported. Come back. There we go. So this is everything I've been talking about. Use this for your study guide, because I do expect you to understand transport, because it'll become more obvious in a moment, not in more detail, why we need it. Okay. And this actually, it's 15 after. I'm going to stop here because the next section is about 3 or 4 slides and it's going to exceed my 5 minutes. So I'm going to stop now. It's 9:15. We will continue on Thursday. All right. So thanks, everybody. I will post the lecture recording in a little bit.